Hello, hi. We were doing the survey of module fungi and we have finished Zygomycota. Today we are starting Ascomycota and Saccharomyces is the first genus which we are going to see. Before moving into today's agenda, let us see. This is the order in which we will be proceeding with the fungi module. We have already covered two important life cycles under Zygomycota, that is Rhizopus and Mucor. In Ascomycota, we will be seeing Saccharomyces, Aspergillus, Penicillium, Neurospora, Pezisa and Claviceps. After that, we will be moving to Basidiomycota. Under this, we will see Puccinia, Agaricus and Stylago. And this, up to these three, like Zygomycota, Asco and Basidio are the true fungi. Then we will see fungi-like organisms called Oomycota. Under the phylum Oomycota, we will see life cycles of Phytophthora and Albugo. Then we will have some general topics and like lichens, mycorrhiza, fungal toxins, etc. So let us move to today's agenda, that is Saccharomyces. It is also known as yeast. Now our agenda will be to study about the general salient features and habitat, nutrition, etc. of Saccharomyces for a short note. Then the structure of yeast or Saccharomyces. Then vegetative reproduction, sexual reproduction and economic importance. So whenever we start writing about a genus, we have to write about the systematic position. It belongs to kingdom fungi or mycota, then division Ascomycota, class Saccharomycetes, order Saccharomycetes, family Saccharomycetaceae, genus Saccharomyces. So this is the body structure. This is a particular stage in the reproduction. You can see budding. We will see how budding happens, etc. And in this image, you can see that as in every other fungal cell, almost all the eukaryotic cell organelles like mitochondria, vacuole, endoplasmic reticulum, Golgi apparatus, ribosomes, everything is present in this particular Saccharomyces fungi as well. So whenever there is a short note about question on structure of Ascomycota or Zygomycota, or if they are specifically asking about one particular genus like Saccharomyces, so what all should be the things that you should write? If they are asking about yeast or Saccharomyces cerevisiae, etc., you can start writing like this. Yeast is a group of unicellular, non-mycelial or pseudo-mycelial Ascomycetes. So what is the uniqueness of this Asco member called Saccharomyces? The unique property is that it is unicellular. In Rhizopus and Mucor, etc., when we saw Zygomycota, we saw that they are having proper mycelium. Okay, there is an erect mycelium called sporangium 4 which will be bearing the sporangium. There is a prostrate mycelium called stolon, and in Rhizopus we saw there is a rhizoids as well. So this is not the case in yeast. It is a unicellular fungi. Okay, you can see that there is a nucleus. Attached to the nucleus there is a big central vacuole called a nuclear vacuole. Then surrounding that there are small vacuoles. Then these black dots are called oil drops or glycogen, which are the storage food materials in yeast. So they are having a egg or ellipsoidal shape. They are found everywhere and commonly found to grow in sugar-rich organic substrata. These kind of things you can write in detail and more about structural detail. See, this is the note I have already explained. You can write or specify the size of a single cell, 3 to 4 micrometer. That's why it is called a microorganism. You cannot see with the naked eye. Okay, so then other things which you can mention is that they reproduce vegetatively or asexually by budding and fission. That is during the favorable circumstances when sufficient sugar substratum is available. Then in unfavorable circumstances they will choose sexual reproduction. And some more detail, cell wall is made up of chitin and it is permeable for nutrient absorption and excretion. So it is through the body it is absorbing the it is through the cell wall it is absorbing the nutrients and it is doing the excretion. Then intracellular enzymes are present like invertase and zymase. Why? These enzymes will be converting sugar into alcohol and carbon dioxide. That's why we are using this in the baking industry, etc. Okay, Saccharomyces is the reason for the fluffiness of the bread, cake, etc. Oh, that will be becoming like fatty in no time. That is because Saccharomyces has converted this sugar and carbon dioxide is released as the byproduct. So that gas release is making it so fluffy. Then flagella and other locomotor organs are absent in this thing. So when we saw the structure, we didn't see any flagella and all. Then other things I have already explained. Now about the structure called pseudomycelium. This pseudomycelium I'll explain when I'm explaining budding. Then nutrition. Nutrition means it is a heterotrophic organism. Okay, it cannot produce its own food because chlorophyll etc. is absent. So it will be leading a saprophytic life cycle. Okay, it will be growing on the top of some sugary medium, and it will be absorbing all the nutrients from that medium. Okay, so second thing, they are chemoorganotrophs. Okay, if they are given some organic compounds, they even does not need any sunlight to grow. So chemically they can collect their food. So that's why it is called chemoorganotrophs. So organic compounds have the source of energy. They need not get sunlight for that, because they are not using chlorophyll etc. for making food. Then they use the sugars like glucose etc. as a source of carbon. Why? They are converting these sugars to alcohol and carbon dioxide through the process of fermentation. And talking about its general habitat, they grow in neutral to slightly acidic pH environment. Then habitat, sugar rich material, okay, on the surface of fruits and berries, even seen in the saps released by plants or the exudates released by the plants. Then in soils and associated with insects, and even in the guts of mammals, and even in deep sea environment you can observe the Saccharomyces. So these details will help you in writing beautiful short notes on Saccharomyces. Then about reproduction, either you can use the terminology of asexual reproduction or vegetative reproduction in the case of yeast, because different sources are giving budding and fission either under vegetative reproduction or under asexual reproduction. And apart from budding and fission, there is one more thing you have to mention, that is the endospore formation, that we will see in the upcoming slides. So on the basis of the asexual or vegetative reproduction, yeast are classified into two. One is budding yeast and second is fission yeast. Those yeast or Saccharomyces species which choose budding for the asexual or vegetative reproduction, that is called budding yeast. Budding yeast is called Zygosaccharomyces. Then second is fission yeast, that is called Schizosaccharomyces. So is there any tip for remembering? Fission, schizo, that kind of mnemonics you can use for remembering. And sexual reproduction we will see in the upcoming video, not in this video. Okay, vegetative reproduction of Saccharomyces. First one, budding. In the case of budding, what will happen? First of all, one thing you have to remember, that budding or asexual reproduction or vegetative reproduction happens during the favorable circumstances where sufficient nutrients or organic substrate is available. Okay, and one thing is that this is the mode of reproduction which is happening maximum times in maximum number of species. Okay, maximum Saccharomyces species go for budding, and in very few number of species go for other fission or any other sexual reproduction kind of stuff. Okay, but sexual reproduction can be done by any species if conditions are unfavorable. But apart from fission, budding is the most favorable thing. That is the point that I wanted to make. Then just imagine how it happens. Just visualize. So this is the parent thalli. It was having a single nucleus and a proper cytoplasm etc. with almost all cell organelles. Then during the time of asexual reproduction or vegetative reproduction, a particular part of the cell wall becomes soft, and the protoplasm of the parent nuclei, parent cell, will be moving towards that particular bulging or small outgrowth which is getting developed on that particular soft cell wall area. Now nuclei divides, all other organs multiplies, and every single organ gets duplicated and moved to the bud, including the daughter nuclei. Now there will be a septum formation at this part and a proper bud is created. Sometimes, very rarely, you can see one phenomenon called pseudomycelium formation. That is, after the formation of bud, this bud is not getting detached from the parent body. Then the daughter bud form another daughter bud. It forms another daughter bud. So a chain of buds are attached. That is called pseudomycelium. So it's not a proper mycelium as we saw in Rhizopus or mucor. It's just a pseudomycelium. This stage will be quickly over, and it will be releasing all daughter cells into separate unicellular Saccharomyces. So it is this budding phenomenon is observed in budding is called Zygosaccharomyces. So this is the phenomenon. I have explained everything. You can pause the video and take the note. These are easy to reproduce diagrams, right? Very easy to reproduce. Then in the case of fission, what happens? We'll see. In the case of fission, fission is called SACCHAROMYCES, and SACCHAROMYCES, we are taking the example of a specific like variant of SACCHAROMYCES, that is SACCHAROMYCES octosporus. In that you can see fission. In that what happens? The parent yeast cell elongates, so its length is increased. Now fission happens by a transverse wall formation in the area, in the middle part. Okay, before the formation of this groove for the formation of a transverse wall, nuclei and the cytoplasm divides and all organelles replicate, so that one copy of each is created. And once before the transverse wall formation completes, so you can see that from the periphery wall is arising, so it is called peripheral wall formation. So from the periphery or centripetal wall formation, okay, from the periphery towards the central wall is forming. So each daughter nuclei will be carrying the same parent nuclei one copy, because the parent nuclei mitotically divided and transferred its content to the daughter nuclei as well, daughter cell as well. So finally the transverse wall thickens, and as a result 2 daughter cells are formed. This is also an easy to reproduce diagram. Then this is the thing. Okay, this was the parent unicellular Saccharomyces. So it got elongated, a transverse wall or septum got formed, and by that time all cell organelles replicated and one copy each went to each daughter cells. Then it will be detached. It will be detached at this point, and new thalli is formed. Now final vegetative or asexual reproduction mode is endospore formation. What happens? Okay, if the surrounding nutrient medium got exhausted, there is no sugar available or glucose available for completing its life cycle, so at that time its life is in danger. So what it will do is its protoplasm divides into 4. Okay, protoplasm divides into 4, and from one single thalli 4 endospores are formed. Okay, its protoplasm, its nuclei actually divides into 4. Then surrounding each nuclei, some protoplasm gets aggregated, and a thick wall is formed surrounding each nuclei, and that will be forming endospores. And when the conditions are favorable, this mother body or the cell wall of the parent cell that gets divided, that gets ruptured, and these endospores will be released outside, and that will be forming new thallus or new unicellular Saccharomyces cell. So I'll show you how this happens. You can see that this was the parent cell. So when it found that conditions are unfavorable, the genetic material and the nucleus, everything gets divided, then what will happen? There will be this splitting or septum formation in between the parent within the parent cell itself. You can see that this was the single genetic material. It got split. It got a copy. It got divided. Then it got divided, and it moved to one apex of this particular thallus, and there was a construction growing centripetally. That means from the periphery towards the center. So it will be finally forming a solid body. Okay, an ovoid or circular cell. It will be forming a daughter cell. Then that will be engulfed by the parent cell. You can see that this parent cell engulfing it, so completely engulfing it, so completely engulfed. Okay, after the endospore is formed, after this daughter nuclei is formed, the mature cell DNA degraded. Okay, actually it will be degrading after the formation of 4 endospores in this manner. Okay, all the mother cell DNA will be that will be remaining, that will be degraded. So in this way, this is 1. Similarly 4 endospore will be formed. Now this endospore, it is not a proper endospore. Now it is just having one particular cell wall of its own, then of its parent. Now this particular both cell wall, that will be deposited by some peptidoglycan material, and it will be called cortex. Okay, now outside this cortex there will be formation of a protein coat as well, extra protein coat as well for protection. Now it can be called a proper endospore. Now when the favorable conditions arise, it will be released, and that will be forming a new endospore. Till then we are So we wind up here. We'll see in the next.